Thank you so much. Yeah. Ladies and gentlemen, thank you very much for participating in today's talk. I have titled it as an invitation to marine mycology. If you saw it very clearly, very minutely, the title was very different. The marine fungi, what's next for India. I have changed the title to an invitation to marine mycology, considering the background of the today's audience. Most of them may not be very well versed with the nuisances or what you call complications involved in the marine mycology. So for the benefit of such audience, I have titled today's presentation as an invitation to marine mycology. Especially, this is an invitation to young researchers from India, some countries, to contribute to marine mycology. The idea behind this presentation is that I think some months back we had an online seminar for one full day, and we had a very esteemed speakers like Dr. Sridhar, Dr. Venkateswara Sarma, Dr. Surya Narayanan, Dr. Patricia Velez from Mexico, Dr. Mark Calaban, and Dr. Nasim Thakur from my colleague from CSIR, and Dr. Samir Damre gave a wonderful presentation on deep sea research. So after that online seminar, we learned a lot. Personally, I have learned a lot. And Rohit Sharma came forward with an idea that we should publish the learnings in the form of a paper. So he started writing the manuscript, and I got involved in this. And Professor Sridhar actually he guided us to complete the manuscript and publish it in the MycoAsia Journal of Indian Fungi. So this is the paper I have been talking about. Sharma, Shenoy, Sridhar, 2024 Insights on Marine Fungal Research in India. So this is the outcome of our learnings from the online seminar on marine fungi, and many of the experts in the field contributed in the form of criticism to our earlier drafts of manuscripts, and we are very thankful to them. And I am very much thankful to Professor Sridhar and Dr. Sharma, who are the co-authors of this paper. So based on this experience and this collaborative spirit, I am presenting today's talk titled An Invitation to Marine Mycology. So what is the plan for today? So first I will give you a brief introduction to oceans. So what are oceans? How are they important in terms of ecosystem services or ecological services? Then I'll come to the definition of marine fungi, and then give some bit about historical perspective. Then I will move towards the marine fungal research in India, and then I will focus on 4 important areas that I consider very important at the present moment. Actually, the one would be the deep sea fungal research. Second would be the importance of marine fungal cultures. Third one would be going beyond culturing and embracing NGS tools to characterize the diversity of marine fungi. The last one would be realizing the full potential of fungi in marine biotechnology. At the end of this talk, I would like to share a 10-point program for advancing marine fungal research in India. Though I will be focusing mostly on Indian scenario, but whatever the lessons, whatever the ideas we are sharing, probably would be applicable to other countries, especially developing countries where marine mycology is not in a great shape. As you know, our oceans cover the 71% of Earth's surface. So as you can see, we have a blue planet, and as you know, the Earth surface is covered by ocean. It's about 2/3 of it, and oceans are very important. And all these oceans, 5 oceans, are interconnected, and we call it as one global ocean. So one global ocean is very important in terms of ecosystem services. Oceans are very important for climate regulation. Oceans host biodiversity and support different kinds of habitats. Oceans actually support food supply. If I have to give an example, 5% of global GDP is contributed by marine food industry, actually. So then oceans support medicinal resources. You may be you may be aware, but you may not be aware that 50% to 80% of the oxygen that we inhale or consume are produced by the phytoplankton present in the oceans, actually. So, of course, tropical forests are important for the oxygen production. More important, I mean, equally important are our oceans, because they contribute to 50 to 80% of the, or 50 to 70% of the oxygen that we inhale, actually. So, oceans have these ecosystem services have economic value. If you convert this all these ecosystem services in terms of dollars or Indian rupees, it's a huge amount. So, of course, oceans are important as cultural and recreational services. We like to go to the beaches and enjoy the view and spend our evenings, and it supports tourism industry. Oceans are important for nutrient cycling, coastal protection, water purification. So this is just to give an idea that oceans are important for various reasons, and some of the reasons I have already told you, actually. And oceans are, the global ocean is estimated to support 2.2 million species, actually. So 2.2 species of different forms of life could be there in our ocean or in the global ocean. So let me read out from this table from Mora et al. 2011. So you know, in the oceans we have around 171,082 animals, then Chromista, then around, I'm talking about 2011 data. That's in 2011, around 1,100 fungal species were already documented from the marine environment. So you find many different species of plants, protozoans, and archaea, bacteria. So many different kinds of life forms are supported by our global oceans. But as per the prediction, there could be as many as 5,320 fungal species in marine environment. And these fungi could be in different forms. These could be mycelial microfungi. These could be single-celled yeast. These could be zoosporic fungi of Chytridium-mycota. Even lichens are present in the coastal environment. Just to give a better idea about the marine fungi, if you look at this picture, you could see different kinds of fungi isolated from one species of sponge. And this sponge is the name of the sponge is Yersinia variabilis. And this image is from the paper published in Fungal Diversity 2010. As you can see, a single species of sponge can support or yield so many different kinds of morphospecies of fungi. So some places in marine ecosystem, fungi can be found in a very different, in many forms and in a rich biodiversity way, actually. Coming to the recent paper by Calaban et al., published in Botanica Marina, they have suggested, they have mentioned that we have so far described 1,898 fungal species from marine environments. Most of them belong to Ascomycota, then comes the Basidiomycota, then Microsporidia, Chytridium-mycota, Micromycota, or some more, actually. So this is about the species, and these marine fungi belong to 767 marine fungal genera and 273 families, 107 orders, and 33 classes. And you can see diversity of taxonomic diversity of marine fungi. So they are diverse, and they are very important. They're important. So, and as you may know, there are two major types of marine fungi. First types are obligate marine fungi. What are obligate? That means they need marine condition to complete their life cycle, and they require salt or saline condition to thrive. So these obligate marine fungi will not be able to survive in non-marine conditions. For example, if you isolate some aspergillus from the deep sea environment, where the pressure is around 500 atmosphere or maybe more, if you try to grow them in the lab condition, it may not grow. So this is an obligate marine fungus, actually. So there is a second kind of marine fungi. So they are called facultative marine fungi. These fungi can grow both in salt water, fresh water, also in marine environment, also on land. So you can find this species in all kinds of ecosystems, marine, terrestrial, estuarine, but they are also found in marine environment, and they are found repeatedly in marine environment. So these facultative marine fungi are known for their adaptability, and they are, for example, best example is aspergillus. You can find aspergillus on the terrestrial condition, estuarine condition, mangroves, coastal condition, coastal waters, open ocean, deep sea sediments. So this is aspergillus is the best example for facultative marine fungus actually. Here I am showing you a recent paper. This is also another example for the obligate marine fungus. So, so this is a discovery of Elysia longicola from deep sea of an island, some islands in Japan. So these researchers introduced some wood samples at the depth of 250, 500, 1,200, 3,000, 5,000 meters. And they removed those wood samples after 3 years or so, 3 years, 7 months. So they could find this kind of beautiful fungus actually. This is an endemic fungus, and this is an, not only this is an obligate marine fungus, this is an endemic fungus actually. So it looks like this is one of the first reports of endemic marine fungi. As I told you, marine fungi are taxonomically diverse, and they play different ecological roles. For example, these marine fungi, they could infect phytoplankton in the marine system. Recently, there has been a report of multi-drug resistant Candida auris of Andaman Islands. So they have reported the presence of this drug-resistant yeast in these of Andaman Islands. We don't know whether we don't know much about their epidemiology. You know, so there is more studies. More studies are required in this direction. Personally, I feel discovery or exploring the drug-resistant fungi in marine environment would be very interesting, especially in coastal environment or in the big tourist beaches. Actually, second type of ecological role played by marine fungi is saprobes. These marine fungi as saprobes, they decompose organic material. They infest or they actually degrade decaying or sponges and zooplankton. And some of these marine fungi are symbionts of seaweeds and other stuff, other life forms. So I was talking about this paper, and probably I encourage people to go through this paper, and you'll find that this is an interesting report from India of Andaman Islands actually. Not talking about the history, because history has defined the present, and the present will define the future. It is better to have an historical perspective about marine mycology. Of course, marine fungi were reported even in 1850s or 70s actually, but the notable contributions came from Sutherland sparrow. Sutherland 1915, Sparrow 1936, then Roth et al. They did the pioneering work in isolating deep sea fungi from depths up to 4,400 meters beneath the sea surface. That's I'm talking about 1964. Then the legends of marine mycology, Colemeyer and Colemeyer, they isolated marine fungi from submerged wood located between 500 to 3,000 meters beneath the sea surface. They started from the temperate regions like Canada or USA. Then they even explored tropical areas like tropical areas of Atlantic, Pacific, and Indian Oceans. This is the paper of Sutherland, where he describes fungi from brown alga actually. Rather than the second paper of Sparrow, where this paper is very interesting. It is freely available. It is about the biological observations on the marine fungi of Woods Hole waters. So this is about waters. This was more about from the intertidal region. This is from the seawater actually. So This paper, this is the abstract of the paper by Roth et al., published in 1964, I guess, 1964, in Canadian Journal of Botany. So this is an interesting, and they described some species of fungi in the, from the marine environment. For example, Oreobasidium pullulans, Dendrophila erinaria. These are the common or normal inhabitants of the marine environment, especially in the deep sea environment, actually. This is a classic book of marine mycology written by Kohlmeier and Kohlmeier. In 1990s, significant advancements were made in marine mycology. For example, Lorenz and Molitoris, Raghukumar and Chandralata Raghukumar and Sheshagiri Raghukumar, my ex-colleagues from CSIR, National Institute of Oceanography in India, they did some wonderful work on exploration of marine fungi, especially from deep sea samples and cultivating them under high pressure conditions in the laboratory. I'm going to talk about deep sea and research in coming slides. My colleague Dr. Samir Damre has did some wonderful work on deep sea fungi, fungi from deep sea sediments from the central Indian Ocean basin, actually. So he studied the culturable fungi from deep sea sediments from central Indian Ocean basin, which is somewhere near Mauritius. And here is a classic book of marine fungi by Dr. Sheshagiri Raghukumar, one of the living legends of marine mycology from India, who contributed significantly not only in understanding marine fungi, in fungus-like organisms like prostochondrids, and also physiology and the biotechnology of it. This is the paper by Lorenz and Molitoris, and here Dr. Raghukumar talking about biotolerance of fungi isolated from deep sea sediments of Indian Ocean. Here is the classic paper by my colleague Samir Damre and first advisor and Dr. Raghukumar, fungi in deep sea sediments of central Indian Ocean basin, actually. So coming about the India's coastline. So India's coastline is as long as 7,516 kilometers. So you can find estuaries, lagoons, mangroves, backwaters, salt marshes, rocky and sandy coasts, and coral reefs. So that means there are great opportunities to explore these places for marine fungi. So these ecosystems or these habitats not only supports fungi, they also support algae, phytoplankton, zooplankton, coral reefs. All right. Talking about prominent Indian researchers who worked on marine fungi, who are still working on marine fungi. Number one, I would be Professor Venkateswara Sarma from Pondicherry University, whom I consider my guru of marine mycology. Whenever I need some advice on marine mycology, I consult him. He is doing wonderful work on marine fungi of India. Then we have this classic book of marine fungi of India by Borse et al. Then Professor Suryanarayanan, Professor Sridhar, co-author of this presentation, Professor Sheshagiri Raghukumar, Dr. Chandralata Raghukumar, Dr. P. Manimohan, my colleague Dr. Samir Damre from CSIR, NIO Goa, Dr. Varada Samir Damre, who is an expert in prostochondrids from Goa University, Dr. B. Devdatta from CSIR, IMTech Chandigarh. So here you can find the photographs of these people, Professor T.S. Suryanarayanan, Professor Sridhar, Dr. P. Manimohan, Dr. B. Devdatta, Dr. Sheshagiri Raghukumar, Dr. Chandralata Raghukumar, Dr. Varada Damre, Dr. Samir Damre. Talking about my organization, which has been involved in the oceanographic research since 1966. It is based in Goa, Indian state of Goa, in a place called Donapaula. Then we have 3 regional centers because we cover 2 coasts, actually big coasts, long coast, west coast of India and the east coast of India. So on the west coast of India, we have 2 regional centers, one at Kochi and another at Mumbai. The 3rd regional center is on the east coast of India at Visakhapatnam. I am working at the regional center, Visakhapatnam of CSIR, National Institute of Oceanography. We have 2 research vessels. One is Sindhu Sankar, mostly for the coastal research, and we have a very beautiful research vessel for deep ocean research or deep sea research, what we call. This is the name of the ship is Sindhu Sadhana. This is the picture of our ship, and it costed us around 400 crores Indian rupees. You can make the calculation in US dollars and other currencies. So this is a beautiful ship. I did this participate in research cruises. I went to Central Indian Ocean Basin in this ship. Actually, then I also went to the Western Arabian Sea for the sampling. So this is a beautiful ship, and lots of sampling has been done using this ship from the deep sea for deep sea research. Marine fungal research in India has been focusing on marine water, driftwoods, mangroves, hydrothermal vents, fishing boats, solar saltans, deep sea. And I've been working on fungi of tadpoles. Not only I have been working on fungi of tadpoles, I have done a good amount of work on the bacteria associated with tadpoles of the west coast of India. As you know, tadpoles are a marine pollutant. I'm going to talk about our work on tadpoles in coming slides. So I'm coming to the core of today's presentation, where I'm going to talk about 4 focal issues. I consider these 4 issues are very important for marine mycology in India. First one would be the deep sea research, or deep sea research, or deep sea fungi. Second one would be about the importance of culturing marine fungi and maintaining a maintaining a collection of them for biotechnological users, application, or for the future generation of India or the world, actually. So third one, I will be talking about going beyond culturing, embracing NGS tools, especially in the Indian context. And I will talk about how to realize the full potential of fungi in marine biotechnology, actually. So let me start with the deep sea fungi. So what is meant by deep sea? Anything below this shelf, you can see shelf here. So that is deep sea. Whatever you find here, we can call them as deep sea fungi, actually. Okay, so it can go as deep as 10,000 meters. That is 10 kilometers below the sea surface, actually. So in our research cruises, in our research endeavors, we have collected samples from 4,000 meters, 5,000 meters, 6,000, almost 7,000 meters from the Central Indian Ocean Basin. Why is it important to study these fungi from this deep sea environment? And as you know, deep sea environment is characterized by low temperatures. Temperature can go up to 4 degrees Celsius, and it's high pressure. So when I'm talking about 5,000 meter depth, I'm talking about 500 atmospheric hydrostatic pressure. That's lots of high amount of pressure. So how do these organisms survive in that kind of an environment? And we have kind of feast and famine nutrient condition. Mostly famine, sometimes feast. So these organisms have to have some adaptations to deal with or to survive or probably thrive in this kind of conditions. So I consider, and many people who are working in the marine mycology in India consider that the deep sea is a fascinating environment for scientific exploration, because of we can get very useful insights on the survival strategies of marine fungi and other life forms in the deep sea environment, which is characterized by low temperatures, high pressure, and feast and famine nutrient condition. And one of the adaptations of these marine fungi could be to produce secondary metabolites or enzymes to deal with the high pressure or low temperature and famine, lack of nutrients, or poor availability of the nutrients. So this could have potential in bioprospecting. As I mentioned, my colleague Dr. Samir Damre did wonderful work on deep sea fungi from Central Indian Ocean Basin. He has collected them, not only isolated them, he grew them in the laboratory condition using pressure vessel, and he did some physiological experiments, try to understand how if we mimic the deep sea conditions, how these fungi will respond, in the laboratory conditions. One of the main challenges for deep sea research is the limited sampling resources. Only a few laboratories in India can afford this kind of sampling resources, sampling logistics, And a ship in India will cost you 1.5 million rupees per day. It's a very expensive research, but interested parties can start talking to CSIR laboratory like NIO, or Ministry of Earth Sciences laboratories like NIOT Chennai for deep sea research. And not only that, there is a need for innovation in sampling of deep sea fungi, actually. Second aspect I wanted to cover is about marine fungal cultures. We have many culture collections of India for fungi, but we don't have a dedicated culture collection for marine fungi. Why do we need a dedicated marine dedicated culture collection for marine fungi of India? Because marine fungi are a different kind of, different kind of entities. They are very crazy. They don't obey. They are not easy to culture. I'll tell you. Normally, we isolate fungi from seawater and other samples, transfer them to high salt media to mimic their natural habitats. If we are isolating fungi from coastal environment, then seawater will be the salinity will be quite high. Then we go to open ocean, and if you go to some other places like estuary and all, so then the salinity will not be that high. So we need to use seawater. We need to use seawater as a growth medium in addition to other other minerals or other components of the culturing. It is important to do the culturing. Of course, there are NGS tools for understanding the diversity of marine fungi, but it is important to culture marine fungi, especially obligate marine fungi, which cannot probably live outside the marine conditions on land, but probably we can make them live for some time in the laboratory conditions. Therefore, we need to understand the nutrient composition of the media that we are going to give to for use for the sampling or the culturing of these marine fungi. Why do we need to maintain these live cultures of marine fungi? For physiological studies, for biochemistry studies, even for industrial applications, we need to maintain cultures. But the major challenges are viability loss during storage, and if you are into some application research, diminished bioactivity after subculturing or something. So what is the way forward? Probably we need to do more research into the preservation methods, cryoprotectants, and novel isolation media. I would probably stress more on the designing more interesting and very effective isolation media for marine fungal research. A national culture collection dedicated to marine fungi probably would be great actually, because it will help in the conservation, taxonomy, and bioprosperity. In a culture collection, I work for culture collection at CSIR MTech in the MTCCs. You don't have experts of everything. There could be one or two mycologists. They are experts in one or two aspects. But marine fungi probably needs more than one mycologist to handle these cultures. So for Prostros, an expert would be needed. For deep sea, another expert would be needed. For the for the chemosynthetic or for different life forms, for different life strategies of these fungi, we need different. So especially if you are really serious about marine mycology research in India, definitely a national culture collection dedicated to marine fungi would be a good idea, actually. So let me talk about the third important point, by embracing next generation sequencing tools. I have been asking my colleagues, Dr. Samir, how many studies have been done on marine fungi that employed amplicon sequencing of ITS region. We have hardly had research on marine fungi using ITS, amplicon sequencing of ITS. Most of the studies have been, culturing them, describing new taxa, doing some kind of biochemical, biophysiological studies, understanding the effects of some ecological factors on the growth, some biotechnological work. But embracing NGS tools hasn't been that much encouraging, actually. So, what I'm trying to say, we have not used NGS tools that much to understand the diversity of marine fungi in Indian waters or Indian deep sea sediments. So I want to explain what we did. We collected in this study published in the MycoAsia Journal of Modern Mycology, in this paper titled Exploring Fungal Diversity and Their Ecological Roles in the Coastal Waters of Ramakrishna Beach, Visakhapatnam, India. We went to a beach on a Sunday evening where there would be thousands of people, on their beach. and we collected seawater. All right, and we collected Ramakrishna Beach is the name of the beach in Vizag, from where we collected the seawater on a very busy evening on a Sunday. And we did the DNA extraction, and we did the kind of amplicon sequencing of the ITS region. Then once we got their diversity data, we predicted ecological function. To our surprise, we found this kind of fungi in our NGS analysis. We found Candida tropicalis, we found Candida hyderabadiensis, we found Aspergillus penicilloides, we find Candida cruzi or Tenomyces cruzi, we found Pleuromyces lactase Candida, one more species, Candida glabrata, Candelomyces, Candoliomyces, and Aspergillus conicus, and one more, a couple of more. So we could find Aspergillus. We could find many Aspergillus species. We could find many yeast species in the coastal water. It looks like this coastal water is highly influenced by anthropogenic activities. And we not only did the taxonomy diversity characterization, we did the prediction of the possible ecological functions using fungal analysis. So we found the Candida tropicalis could be pathotroph, saprotroph, symbiotrophs. It does not get we can have a very dynamic lifestyle actually. For the second species, we did not have much information in the fungal database, that is Candida hyderabadiensis. For Aspergillus penicilloides is a pathotroph. It is an animal pathogen. Interesting to detect it in coastal waters of Visakhapatnam, Ramakrishna Beach. Then we have some more species for which we did not get enough data from the fungal, but a fungal database. But still, I feel this is a way forward in addition to the culture-centric studies. The second study is from the Goa, which is on the west coast of India. We have been working on the tar ball samples. Tar balls are nothing but marine pollutants. These are found in the open ocean. When an oil spill event happens, when the oil is introduced to the ocean, the oil slick is formed because of the wave action and other effects. Ball-like structures are formed during pre-monsoon and post-monsoon season. These tar balls, or the ball-like structure of what you call weathered crude oil, they come onto the beaches of, especially onto the tourist beaches of Goa. And when these tar balls come onto these beaches, especially during the tourist season, it is not a good so the tourist department of the Goa manually removes them and dump these tar balls somewhere else, and so that the beach is clean. But what we have been doing, we have been trying to understand the microbes associated with tar balls. We have been studying the bacteria associated with tar balls from some beaches of Goa. We have also studied the tar ball fungi associated with tar ball samples on Goa. So we did the culturable studies, then we followed it up with the NGS analysis. We collected tar ball samples from two beaches of Goa, Vegator and Morching, and we did the amplicon sequencing of ITS region. When we did predict the ecological functions, so this is how the result looks. We found so many Aspergillus species in both the beaches, and when we did the ecological prediction in the fungi, we could find pathotrophs, saprotrophs in both the beaches. No symbiotrophs, because tar ball is a marine pollutant, and it's like I don't think we can expect symbiotrophs on a marine pollutant. The last point I would like to highlight is the fungi in marine biotechnology. So secondary metabolites have been extracted from the marine fungal cultures, which includes steroids, alkaloids, terpenoids, which have been shown to have anti-tumor properties, anti-cancer properties, antimicrobial properties, antioxidative properties, anti-inflammatory properties. It seems over 1,000 natural secondary metabolites from marine-derived fungi or facultative marine fungi are examined so far. Only a small fraction of obligate marine fungal have been explored for the potential. Basically, there's a great scope for exploring deep sea fungi for their biotechnological potential. Of course, some work has been done. We can do more than that, actually. And Dr. Raghu Kumar has done wonderful work on prostocrites and exploited these for omega-3 PUFA, and these PUFA, omega-3 PUFA is very important for the human health, also has applications in aquaculture. And this technology is still available for commercialization from CSIR and Goa, where Dr. Raghu Kumar did his research. We see a great potential in using marine fungi in plastic degradation and cosmeceuticals. Recently, we came across one interesting paper about degradation of plastic by a marine fungus. So we are going to see such reports or more repeatedly in coming months and years. So, of course, Indian mycologists have been working on coastal ecosystems because it's easy to access these habitats. It's less expensive. It's important to continue the studies on marine fungi from coastal environment of India because this is important in terms of healthcare, conservation, and industrial advancement. Government of India under the Ministry of Earth Sciences has started Deep Ocean Mission, or DOM. There are many components under this mission. One of them actually is to unlock the mysteries of deep sea. And me and my colleague, Dr. Samir Damre, and my another colleague, Dr. Nasim Thakur, are very excited that we will be spending some years, next 2-3 years, understanding the life of under deep sea environment and their possible application in biotechnology. It's a big project of many CSIR laboratories, including CSIR, IMTech Chandigarh, CSIR, CDRI, Lucknow. Basically, we need to do many more studies in the marine fungal and mycology for India, as there have been very limited studies, and we need more people. Therefore, the title of the presentation has been an invitation to marine mycology, especially to the Indian researchers, budding scientists or budding mycologists. Let me come to a very important aspect of today's presentation, a 10-point program for advancing marine fungal research in India. I'll take some 5 more minutes to talk about it. First, we need to enhance the existing research facilities to support advanced studies. University systems, even some research institutes need to enhance the existing research facilities to support advanced studies in marine mycology. We need to probably acquire state-of-the-art equipment for deep sea sampling, for high pressure culturing, molecular analysis, because high pressure culturing is a very the skill is not very much found in many laboratories. A couple of laboratories are working on high pressure culturing of fungi and bacteria. And as I already explained, we need to have a national repository and a database for marine fungi, which that database could include information on the species discovered so far, their genetic data, their metabolites, and their ecological importance. The third is we need to secure more funding for marine mycology, especially for exploration of biodiversity and for biotechnological applications. We need to collaborate more. I've been discussing this with my friends in mycology. We need to do interdisciplinary research. We need to collaborate international experts in different fields like taxonomy, ecology, marine, molecular biology, and biotechnology. We need to address some complex questions. Especially one of the questions is that how is that the same species of Aspergillus can survive in both coastal ecosystem and deep sea environment and terrestrial environment, and even in the air? Then here we probably can use NGS tools or comparative genomics, whole genome sequence data, and other omics tools, and also physiological experiments to address these complex questions. Actually, we need to do capacity building and training. We need to develop specialized training programs and workshops for early career scientists. We have started a WhatsApp group for marine mycologists. So you are welcome to join that group. We need to use modern techniques, and young people are to be trained in these techniques like next generation sequencing and analysis, bioinformatic analysis, especially culturing under extreme conditions. Many more people should be trained, and they should start using this skill for the culturing of deep sea fungi in the laboratory condition. It would be nice we can encourage collaborative projects between academic institutions and industries. I mean, probably one of the ways to do is to keep talking to the industrial potential partners. Tell them how beautiful are these fungi or life forms found in deep sea environment, the kind of environment they are in, they deal with every day. They're producing secondary metabolites, enzymes, a very interesting cell membrane system to deal with the poor nutrient condition, high pressure, and low temperature. Probably this kind of lifestyle, or especially what you call extreme environment, is expected to result in some interesting biomolecules that could have applications in pharmaceuticals, agriculture, bioremediation maybe. So, and it would be good to have collaboration between industry and academia in marine mycology. We need to spend more time on biodiversity exploration because we hardly know anything. We know around 1,900 1,900 species of fungi from marine environment. If you believe that estimate of 5,000 5,000 species of marine fungi to be discovered from the global ocean, still discovering 3,000 new species of marine fungi would take some time actually. And that will take lots of efforts also. But it's important to continue the work to discover new species, not only discover them from deep sea environment, mangroves and coral reefs and many other habitats. You need to understand their roles in marine ecosystems, and that will add value. Just describing a new species probably will not be appreciated much nowadays, because I work in a multidisciplinary organization where I deal with ecologists, biotechnologists. I deal with zooplankton, phytoplankton, biogeochemistry. They keep asking the same question. You discover new fungal species. What is next? And especially ecologists would like to know what kind of roles these fungi play. Because in open and waters, in sea water or the oceanic water, you don't find that many number of fungi, filamentous fungi. You might find yeast, but filamentous fungi are not to be found in marine waters compared to the number of bacteria that we find in these waters actually. But we need to use more. We need to use different techniques, not only culturing. We need to use NGOs or other stuff, and maybe some of these yeast species present in the oceanic water or coastal waters are playing important roles, even in the water system. Of course, fungi play important roles in deep sea sediments. They play important in the degradation of organic matter and other stuff. Last one would be innovative preservation techniques. Probably we need to collaborate with chemistry people, chemical engineers, who are better in designing these preservation techniques and preservation methods. We need innovation in preservation, because large-scale preservation of marine fungi, especially obligate marine fungi, is a different ballgame. So we need to spend some time and some energy on this issue. Biotechnological, pharmaceutical research, biodiversity finally has to have some application. Of course, taxonomy, I'm a taxonomist. I say taxonomy has lots of application. Identification is the practical application of taxonomic research. And whatever the language you use in taxonomy, it is used widely, in the biological research. Whatever the names we propose, whatever the new names, we enrich the language of biologicals. We enrich the language of communication in biological research. But it cannot be just that. We need to collaborate with the other experts for the biotechnological and pharmaceutical research, for medicine, agriculture, and various industries. Probably for antimicrobial molecules, anti-cancer, anti-inflammatory, and industrially relevant enzymes, especially from deep sea environment. Excuse me. The last point. See, climate change is a very hot topic. So is the conservation actually. So we need to integrate marine fungal research into broader environmental monitoring and conservative strategies. I find there would be interesting, there would be many opportunities and funding opportunities to assess the impact of climate change on marine fungal diversity, especially in mangrove systems. Because mangroves are very precious and the fungi play a very important role there. And what would be the possible impact of climate change on marine fungal diversity in the mangrove systems and other systems also? So we can also focus on understanding the potential of fungi in ecosystem restoration, pollution mitigation. So we need to come out of this mycology shell, and we should try to become a biological oceanographer or kind of a biologist rather than just a mycologist. So as I would like to conclude with that, fungi can feed us, fungi can heal us, fungi can free us, fungi can save our world, and fungi shape our world. With this, I would like to thank Dr. Rohit Sharma, Professor Sridhar, and my employer, Dr. CSIR NIU, my students and collaborators, and my esteemed colleague, Dr. Esami Damre, who is an expert of deep sea fungi, and the many experts who commented and gave suggestions to improve the manuscript, Dr. Manjusha, Dr. Deshmukh, Dr. Jeevan, Rajesh Jeevan, Dr. Suri, Dr. Jairam Bhatt, Professor V.V. Sharma, Dr. Patricia, Dr. Rajesh Kumar, Dr. Varda Damre, Dr. Devdatta. I am very much grateful to MycoAsia and MycoIndia community for their continuous support or constant support. And I am very much thankful to today's audience for your patience and participating in today's talk. These are my contact details. If you don't know me, if you don't know my contact details, you can find me on WhatsApp. You just can WhatsApp to this number, and you can email me also. With this, I conclude my presentation. Over to you, Dr. Rohit Sharma.
Thank you, Dr. Shenoy. Thank you for a very nice talk. I'll keep my words for the end. I'll before others ask their questions, I have a couple of from my side. Are there any kind of, like you was telling that in the presentation, that there are when people can collaborate and write to NIO and kind of collaborate for the sampling kind of thing. So is there any annual call that comes for the students and youngsters where they can apply for the thing?
Not from the NIO, but NCPOR, one of the MOS labs, they take researchers to the North Pole and South Pole for the research there, actually. So there is an annual call for that. And for CSIR, people can collaborate under different invitation funding opportunities, actually. There is no annual call.
Oh, okay.
From CSIR and NIO yet. But this is a good suggestion. I will take up that with the concerned authorities. Thank you for that.
Yes. And just as one small question, that is there, how important is the shock that is taking place from the marine environment to the media plate in case of marine fungi?
They just cannot grow.
Yeah.
It's like you go to, we are living at 1 atmosphere pressure. It's like we cannot go beyond 50 meters, actually. The diver is also 50 to 100 meters, not more than that, actually. So you're talking about 500 atmosphere pressure is, for human being, is like, you will just vanish in a minute or seconds, actually. For the fungi to survive in laboratory temperature from 500 atmosphere pressure into 100, that's a great shock, actually. But if you mimic the deep sea environment condition, somebody uses pressure vessel in the laboratory, so where he maintains the temperature of, pressure of whatever the pressure, 500 atmosphere or whatever maybe, and the temperature probably also. So we can maintain them for some days, actually, or some months, if we keep on subculturing them, or we need to give them condition they were originally isolated from. Yeah.
Okay. Thank you. Professor Sridhar sir, please ask your question. Professor Sridhar sir, please ask your question. Oh, it's not audible.
Wait. Professor Sridhar, can you hear us? Dr. Sharma, can you hear us?
Yeah, I can hear you.
Okay, thank you. I think there is some technical issue with the Professor Sridhar. Any more questions?
I think he has to unmute or
I think.
Anyhow, can I have some observations?
Yes, please.
It's a wonderful lecture, and I have to learn that you have shown a roadmap for the future research on marine fungi, and I've been advocating for culture collection for marine fungi for a long time through some of the articles, and the good that you have brought it up once again with the facilities and the capacity that NIVO has. I think it is ideal that NIVO can take up the culture collection for marine fungi, uh, whether it be at Goa or any other center of NIVO. And good that you are getting some funds for deep sea fungal research. What I felt is that mangroves, we are getting a lot of new fungi. My exploration shows that every time we explore new areas, we are getting some good new fungi. So, how about going for this environmental type DNA sequencing type of studies in mangroves with applying your NGS and metagenomics, etc.? Because from mangroves, you have identified fungi to a larger extent. The already some of them have already been sequenced for ITS and also some multi-gene sequence. So through your environmental sampling, if you get some of those matching sequence data, it would be interesting to see the diversity, whether it is going to be just a usual Aspergillus penicilli, or you'll also be getting the typical marine fungi from these mangroves if you go for the environmental sampling. So the similar type of protocols or techniques that we adapt for deep sea fungi may be replicated in mangrove sedimental samples or any of the litter samples that you may process. That would be interesting, I think, but depends on the funds availability and the people available for that kind of research. And as far as the comment on taxonomy, the marine fungi are very slow growers. So of course, we report something as new, but then growing and establishing the fungus itself is very difficult task. And invariably they don't produce the sexual structures, even asexual structures or anamorphic states getting in the cultures is very rare. So you may end up getting hyphal states, and those marine fungi, particularly the obligate ones, are very slow growers. So exploring the biotechnological potential, whether in the form of enzymes or metabolites, is somewhat difficult. Maybe you will get a clue if you can grow the such obligate marine fungi. You can screen and get some compound, if it is a novel compound, but you cannot grow it at large scale and then produce that novel compound. Maybe you have to either take it through genomic route to some bacterial system, or you have to go through the chemosynthetic route where the chemist can combine those combinations and then produce the drug. But whatever may be the case, the marine fungi, obligate or otherwise, they will give you a clue about the structures, novel compounds. These are my some of the observations. Glad that you have some ideas how to take it about for future marine fungal research. Thank you.
Thank you, Professor Sarvagaba. First of all, thank you very much for attending this talk and giving your opinion and expert advice. First of all, about mangroves. Of course, we would like to work on mangroves using NGOs and more than that. I think Dr. Varda has got some funding recently from Goa government for understand the mangrove fungi and prostocryptids. So she will be doing some things from Goan mangroves. I'm interested to work on mangroves too, but at the moment, as I'm involved in deep sea research under this deep ocean mission, I'll be focusing more on the deep ocean mission project, and I'll be trying to. But there is an aspect in my research is I would like to compare the whole genome of Aspergillus, for example, from isolated from estuary or coastal environment like beach or coastal waters, and compare it with the isolated same species from the deep sea environment. We would like to do comparative genomics in trying to understand at the genetic level what are the differences. How probably there that might give some clue about the adaptation. When I am doing this comparative genomics, probably I will also consider the mangroves as one of the possible sampling sites. Second thing about the, I think you talked about the NGS. I think second one was the, what was the second one point, Dr. Professor Samagaram?
Yeah, I just want to see I'm interested in seeing the diversity of environmental sampling of the DNA and the hits that you get, signatures that you get in comparison to the regular woody substrata, their examination, ITS of the cultures. Just want to see the matching because whether our routine studies of mangrove fungi through direct examination and isolating cultures will help in matching or identifying those environmental fungi, means the samples and the fungi that we get through those NGS studies.
Yeah, as I told you, this is an interesting thing to do. It would be interesting to do this because I talked to Dr. Samir. I asked him how many studies have been done using amplicon sequencing of ITS for marine fungi. He said almost nothing actually from India. And then I had to project my work of tar balls and the seawater from the Ramakrishna beach. So this is a good positioning of the research, my own research. That doesn't mean it is the end of it. We would like to do some more work on mangrove fungi, as Professor Sharma suggested. You talked about one more thing, Professor Sharma. I just did not catch it that time. Can you repeat that second point?
Second point is the slow growing nature of the obligate marine fungi. How are you going to tackle
That's a good
exploring for biotechnology potential enzymes or metabolites? They're going to be very slow growing and it's going to be a big challenge.
As you know, even bacteria, it is estimated that we can culture only 1% of that. Only 1% of the estimated bacterial species could be cultured because culturing cannot be effective for all types of environment. So I think it applies to marine fungi also, especially obligate fungi, especially from the deep sea environment. I had spent some time with Samir Damre on research during the research cruises. I'm just trying to understand that culturing of these deep sea organisms is a different ball game. It is very highly skilled job. It's not a routine culturing. Even if you culture that using a pressure vessel, how do you maintain it? Then we said not only we try to culture, once we culture them, we just extract the DNA and we'll develop a DNA bank. So once we have a DNA bank of these precious different cultures, fungal cultures, probably somebody will do whole genome sequencing, or we will, if you have money, we will do the whole genome sequencing of it. Once we have the whole genome sequence of it, then we will know the possible genes involved in the production of enzymes, secondary metabolites. Culturing cannot be the solution for most of the biotechnological applications. Genomics route and the omics route will have to be taken in addition to culturing. Professor Sharma.
Yeah, even in the case of this marine fungi from mangroves, many of them are slow growers. So that problem is there. And they were culture collection center for marine fungi. I think NIO Goa is well suited for that. And I hope NIO will think about that, your headquarters. That's a good idea for marine fungal culture collection center.
We have already, thank you for that. I have already discussed that issue with Samir Damre. And one of the problems is the establishment of the liquid nitrogen facility. So
and 15 days you have to change every day. 15 days you have to change it.
Yeah, even if you get money, probably that establishing a that liquid nitrogen facility will be quite challenging in that campus. So we have thought about it, but I think.
But at least the initial processing you can do it there, and then later on you can submit to Chandigarh MTCC or NCCS or Agarkar Research Institute. At least for initial processing, it will be helpful.
And another thing is there has to be some network of marine mycologists and the culture collections of India. Actually, probably many of the people are working on marine fungi, and if they try to deposit in NFCC or MTCC, unless and until it's a novel species or novel taxa or some biotechnologically important culture, they may not take routine cultures. But what will happen to these cultures? Probably some of them are interesting. We don't know. So because maintaining a culture in a culture collection is a very expensive affair actually. So so that will also cost we'll have to consider, I guess. But thank you, Professor Sharma. It's nice interacting with you.
Thank you.
Yes. I like to just add a few points, Dr. Shenoy, like what Dr. Sharma sir was saying, that I think the comparison of the whole genome, as you said, will definitely give a lot of information on the adaptations thing, definitely. And I think the preservation of bioresource should also be considered instead, along with the whole genome, like genomic DNA kind of thing. So bioresource centers are complete, the sample from the marine sample should be completely preserved, so that the complete thing is preserved. And definitely, as you said, that marine fungi will be losing some of its biotechnological properties on subculturing. So as Dr. Sharma sir was saying that it needs to be taken for the biotechnological potential, it needs to be explored for its transfer of the bioactive compound or bioactive gene into some other thing and then explore kind of thing.
The thing is, I think, I don't know, some Dr. Professor Sharma and these hardcore marine mycologists. I am a, what you call, facultative marine mycologist. I am not an obligate marine mycologist because I have worked on terrestrial fungi, plant pathology, plant pathogens, insect pathogens, and airborne fungi. So I am a facultative marine mycologist, but these hardcore, obligate kind of marine mycology, they might have studied, isolation methods, and in spite of their efforts, they say that it is not easy to culture these marine fungi. So, but their expertise should be utilized to come up with new isolation methods. That's what I feel.
I think Devadatta would like to join. He will give his inputs also.
Devadatta, please. Yeah.
Yes, sir, it's a nice presentation, sir. I mean, for the future of Indian mycology, and especially you're targeting about marine fungal culture collection center or marine bacterial culture collection center will be really great thing, great idea. Actually, Professor Sharma already wrote about that. I also read at the time. So actually, my question is like you have discussed about amplicon sequencing on marine fungi, right? Which you have collected from Visakhapatnam, RK Beach or some other areas. I wanted to know like how extent we have that reference sequence data for comparison of this marine fungi in the fungal or any other databases.
Oh no, I mean we don't see this. I think our work on tarball fungi, NGS work, ITS amplicon sequencing work, and the Ramakrishna Beach work. I think these are probably these are the two studies from India. That's for us on. That's what I remember. I asked Samir, are there any more studies? Because when we were writing the paper, we wanted to compare because the reviewers asked us to compare with the known things. But there were not many known things from India, of course, from the global things. But global thing comparison is a different ballgame. So we need to generate more data. See, that Ramakrishna Beach sample was one sample data. But still interesting, one sample data is not enough. For tarballs also, two tarball samples. We need to do more sampling and do the generate ITS sequence data. So what is your specific question there, Datta?
Sir, we have very good reference sequence databases for amplicon sequencing and all this, because after we generate the data, right? So in which databases can you please suggest some good databases? I'm also in the process of doing some amplicon sequencing.
So I think what we did, we are also not experts. Okay, I mean we did extract DNA, we sent it to by Europeans. They did the analysis using Unite database. Okay. Unite data, but one of your students have to learn how to do it herself or himself, whatever, Let's say now it has become a very routine thing in China and other parts of the world. Only we Indians have to catch up now.
Yes, sir. Thank you very much, sir. I also experienced the same problem that we also face when we are working with marine fungal metabolites and from true marine fungi, obligate marine fungi, we also lose the vitality during the course of time. That is a major problem we had.
I think it is not only a problem in the marine fungal cultures, even other cultures we have the problem. Probably not that great extent actually. But we have problems, viability issues in our drug discovery experiment projects actually. If you talk to Dr. Thakur, he will explain this problem very clearly.
Thank you, sir, for your
Thank you. Dr. Professor Sridhar, you have any question?
Yeah, can you hear me?
Yes, sir. Yes, sir.
Yeah, I'm very happy that you have projected very nicely for the purpose of beginners and also those who are interested in marine fungi. To add some information, see, all of us are not in a position to go to ocean and collect samples and all such things. But although there is some restrictions and some occasionally we may find that opportunity. But nevertheless, as somebody Sharma mentioned, we can get a lot of. We are accessible for mangroves. without any problem. And then we also accessible for the coastal sand dunes or coastal region. In the coastal region and also mangrove region, we can get several substrates like woody material, seaweeds, seagrasses, live animals, and also dead animal substrates and such things. So those are all some potential material for isolation of fungi. They are maybe some of them may be terrestrial, but you may get some typical marine fungi for isolation and for further purposes, isolation and culturing, such type of thing. That's all. That's what I wanted to mention for the beginners. Those who are interested, they can go and collect the samples and sediments at different depths, and such things can be done. But to start with, the coastal region will be wonderful place to select some of the substrates for their research. Yeah, that's all.
Thank you very much. Thank you, Professor Sridhar, for that. See, one of the reasons I focused on tar balls and Ramakrishna Beach seawater sampling for my NGS study, because I don't need a ship to go there,
Okay.
And if you work on marine pollutants, that will get lots of visibility compared to you go to some part of the ocean, you do some isolation.
Yeah.
Because especially that's what my colleagues told me, that if you work on marine pollution related issues, that will give you lots of visibility.
Yeah.
So if you see my papers, both of them are related to pollution or indirectly related to pollution. But as you said, this kind of collecting sand, arenicolous fungi that we discussed other day, mangrove woods, rhizosphere system, and others, especially impact of climate change on the mangrove system. Mangrove rhizosphere.
Another possibility is also we can visit some of the small islands close to the coast. So that is not difficult because, for example, a Karwar region, we can find several of them. Some of them are restricted, but other the islands we can visit and collect samples. That's what I did to start with marine fungi with Prasanna Rai in early 90s. Then he did some work on some of the islands near Karwar like that. And we can find, I think there is a lot of importance nowadays. India is giving, that is, government is giving a lot of importance for the islands, in the coastal islands, their developments and like that. So such exploration will be of some use for us. Thank you.
Thank you, sir. What I feel, sir, we have to probably change first of all, number of people working on marine fungi is reducing. You look at my NIO. People have changed from marine mycology to bacteriology, seaweeds. Yeah. And some people who are experts in marine mycology, they are in a different institute, they are working on probably different group of organisms. So the expert, even the people have been trained for marine mycology, they are not in a position to do marine mycology. That is it's a little bit concern, actually. So we need to get more manpower. We need to get more projects under this project.
Yes, correct.
When we write proposal, if we just write taxonomy and ecology, the funding agency has not much excitement about such proposals now. We need to probably collaborate with biotechnologists, chemistry people. What you call multidisciplinary thing would be raised, especially under deep ocean mission.
Yeah.
We got funding where some 10-15 CSIR labs are involved, and different kinds of expertise are involved. Taxonomy, so to start with, ecologists, biotechnologists, drug discovery people. We're waiting to see the outcome of this project, though. Thank you, Professor.
Thank you, Dr. Shenoy. Thank you.
Thank you. Professor Sarma?
Yeah. In Devadatta studies, when we were exploring this marine fungi from Muthupet, we encountered some medically important fungi also. And we published a paper asking a question whether mangroves serve as reservoirs for those medically important fungi, because some of them are known to be human skin disease causing fungi. And of course, there in the mangrove environment, they seem to be saprophytic or maybe opportunistic at the most, but potentially they have a potential to become virulent pathogens also. We don't know, but Ours is a preliminary study, but we did ask a question whether mangroves is serving as a reservoir for some of these fungal disease-causing strains. That is my first observation. Second one is like this Antarctica expedition, where they invite other institutions to join them to go and work in the Antarctica. Is these cruises and the ships that you have from NAO, are they open for other institutions? Of course, merit-based proposals or any of the studies.
See, recently we got this funding under Deep Ocean Mission. Okay, so our CSIR partners, CSIR MTech people can come for sampling. So there are, sorry?
Parochial and biased approach.
No. This is no. Listen to me. What I'm trying to say.
Okay.
So this costs money. Each ship cost you 15 lakh rupees. Okay.
Okay.
So if every university person says, I want to come and do the, first of all, they should have expertise for deep sea culturing.
That's correct.
They have to be trained. If you are already trained and you have money, you are welcome to join the project. For the cruises. Problem is, you say, I want to come for a sampling, and you will give me the sample probably. I don't know how to culture deep sea fungi. So it is not a nice situation, I believe. So some training has to be given about the deep sea culturing, I guess. About the MOES labs, they take you to this land there actually, and they collect. And how deep they go, I don't know. In the North and South Pole. How do they do deep sea sampling there in the North and South Pole? I don't know.
No, they just ice.
Yeah. But I think, see, Dr. Sarma, the money is the main constraint there, actually. So the ship, I mean, our ship is not only for the deep sea research. This other is taken for the geologic study, physical oceanography study. Getting ship time is not easy, and it is very expensive. If you are taking that ship for 30 days out, that will cost you 5 crore rupees. Who has 5 crore rupees in Indian marine mycology?
That's correct.
So it is, they will say, but I think the funding agencies have to encourage participation from the small, university people and all. Of course, I think under this Deep Ocean Mission, we are getting MTech people and other people for sampling with us actually. And then because that is a project funded only for the CSIR laboratories.
No, not I am talking in terms of project, but as a policy, as cruises, whether they are open for.
Cruises is the main concern is the cost, and even when we got the funding, they say one of the cruises you take because we are already funding you in another project, in PMN project. So the cruises are not easy to get because cruises are very expensive. You know that, Professor Sarma.
That's correct. Yeah. It has to be indented in a individual project also.
You see, if you think anybody will get 5 crore rupees for the ship cost, in an individual project, if you are taking out for a deep sea ocean, you would need at least 25 days, right? Even within that 25 days, it would be like 3 crore rupees. Which funding agency will give you 3 crore rupees for an individual project? It has to be multidisciplinary, multi-institutional. That's what the approach has been.
Maybe some travel come bench fees they can charge.
Problem is, NIA is mandated to do oceanographic research, but the universities can join. But the mechanism has to be kind of discussed, and there has to be streamlined, I guess. But this has to be done for the betterment of marine mycology in India. I agree with you that.
Oh, thank you.
Thank you, Professor Bhatt, please.
Yeah, Damodar, you gave a very nice talk, Damodar. Very nice. Wonderful talk it was.
Thank you, sir. Thank you, sir.
See, we were discussing about this, some of those human pathogenic fungi occurring in the marine coasts, then mangroves and so on, right? I think we are concluding what's happening. We shouldn't come to a conclusion so fast. I saw your paper on Ramakrishna Beach, where you said Candida species, aspergillus. I think they are bound to come. We really don't know why they are there. Similarly, in mangroves, Professor Sharma said, we see more of these medically important fungi. I will give an example. See, long ago, in 1981-82, when I was working in Ethiopia, the Massawa coast, in the Red Sea coast, in Massawa. I had a student working on marine and mangrove fungi. Mainly, he worked on the intertidal driftwoods. We collected Saxenia vasiformis on an intertidal wood. Now, if you look at this fungus, it was originally collected in 1950s from a forest in Madhya Pradesh by Professor Saxena. Now, subsequently, this fungus, Professor Gareth Jones collected it in this fungus in somewhere in Philippines coast, then from Japanese coast, then in Mangalore, we collected it on rice grains. So these fungi, Saxenia vasiformis, is also known to be a human pathogen. So it occurs in as a saprophyte on wood and marine coast. We don't know where else. So fungi will appear everywhere. Then why it is appearing? I think that should be kept open for future studies. So, for example, Candida species. Many of them, they are there if you can always isolate them. So we should keep our mind open. This should be the message, with all of us. Why they are appearing like that? We still don't know, but we conclude before we collect them, then we isolate and we say, oh, there's a preponderance of these fungi in the coast, maybe anthropogenic effect. We still don't know. We should put a question mark and give some observations. That's a suggestion for future studies. Thanks a lot. Wonderful. You spoke well, and those 10-point future plans are really good. I say really good.
Thank you.
Keep it up.
Thank you, Professor. But about there's a definition of marine. What is a marine? Obligate marine fungi. There is a definition for facultative. There is a definition for marine derived. I would just say the fungi. Find fungi found in marine environment. That's it.
Correct. Yes.
I would just start with that. Statement. Fungi found in marine environment. See, the same species of Aspergillus can be found on the probably on terrestrial environment, in the estuary, freshwater, Ramakrishna beaches, seawater, and the sediments of deep sea. That Dr. Samir has found some of the Aspergillus.
Yes.
So I have trying to recruit a couple of people on the DBT-RA call, where I would like to do comparative genomic studies, not only comparative genomics, physiological studies. How come members belonging to the same species are found on different environments, marine environment, coastal environment, deep sea environment, coastal environment, mangrove environment, terrestrial, and maybe air? How these members are capable? Are they really surviving there? Are they really flourishing there? What is it? Lots of interesting questions can be asked.
Yes.
Some of them could be answered, but only thing is we need more people actively working, because some people got diverted into marine bacteriology. Now coming back to the marine mycology. So I see a great opportunity, and this is a kind of indication for my own personal ambition in marine mycology. This talk. Thank you.
Wonderful. Yeah, it's a good talk. Very nice. I enjoyed hearing you.
Thank you.
Thanks a lot.
Just Dr. Belle, is it okay if I talk a few words?
Please.
See, I to support Professor Bhatt. Actually, we did some work on endophytic fungi of coastal sand dune legumes, and also mangroves of Netravati and nearby mangroves of Mangalore coast. So then we published our papers in Canadian Journal of Microbiology and also Canadian Journal of Botany. And as I am not a plant pathologist, after some time, I come to know that there are some real plant pathogens among the endophytes what we have isolated from the mangroves as well as coastal sand legumes. Then we took the number of this thing, and discussed with the experts, and we come to know that more than half a dozen plant pathogenic fungi are typical endophytes in mangrove plant roots, and also the coastal sand dune legume roots, like that. So it is really surprising to see such type of things, because sometimes because of our knowledge is a little bit constraint with regard to our knowledge, sometimes we may overlook some of these things like that. So Dr. Bhatt has pointed out very nicely about the preponderance of fungi in different ecosystems. So I'll agree with him.
Yeah, I think we should keep our open, and it's a great learning. It's all whole, it's a lifelong.
Actually, I sent some list of species to Dr. Shishupala. He's being a plant pathologist. He has found many. These are all plant pathogens, like that. And also, he has given some information about that. That's how it is sort of learning from different sources, different personnel, like that. Thank you.
At least 4 medically important fungi we have recorded. One is Medicopsis romiroi and Pseudosporium 3 species. All are implicated as human pathogens, and they have been isolated from mangroves. In fact, the sexual state also we got, in addition to the asexual state. Very interesting, and we were wondering whether the mangroves serves as a reservoir for these.
Yeah, but Sharma, you see, Venkatesh, my query is, are there any reports of these infecting from mangroves? Do we have any recorded such reports?
Actually, we collaborated with one medical college, and it showed some preliminary study only. It does have some potential to cause diseases, but not.
No, that's right. But what I am asking is, are there any reports of the infection from mangroves, mangrove habitat?
I don't think so. These are reporting studies, taxonomic studies only.
Yeah. Because this is what area of great interest for future.
Yes, and the overwintering nature is whether they are in the saprophytic nature in the mangroves, or do they cause any diseases on the animals is also not known. It is just overwintering.
That's another interesting area. These are they washed away to the coastal area from the sea, whether they are from the animal source? We still don't know.
Yeah,
any of these things. But it's an interesting observation, and I am really surprised to see, that Ramakrishna beats so many fungi, human pathogenic ones. Interesting, it is really interesting.
Yes.
Yeah. Good. Nice learning.
Yeah, so
Thanks a
Professor,
lot.
I would like to add that Ramakrishna work has to be supplemented. That just with the culturing, see, we might have found the DNA signatures in the seawater of our Candida and all, but we have to do some proper culturing work and to prove that these are Candida actually present. The living population is there, not just the DNA.
Correct.
And that is possibly it is going to be cultured. I think Candida is a common thing in seawater, especially coastal area. But if we get some drug-resistant Candida auris, that will be a big concern actually.
Correct.
And as you know, we can do many things under the banner of marine mycology. When I joined NIO, Dr. Ramaya, my HOD, told Professor Dr. Chandralata Raghukumar worked extensively on the pathogens, fungal pathogens of corals. I think that's what she told me.
Yes.
Yeah. So if you work on this kind of hot kind of topics and marine pollutants, and pathogens, as Professor Shidhar told, mangrove plants, or those are some ways we can attract funding agencies' attention.
Yeah, interesting. Very nice. Thanks a lot.
Thank you.
I just want to make a small point, sir. I think will a reference site will be helpful for the connecting these pathogenic fungi with the anthropogenic activity as a reference site where there is no anthropogenic activity?
Reference center, I don't know, because there has to be some baseline data, right? And who has generated the baseline data for? I mean, one of the proposals we have in mind is, let me just reveal my secrets in the public domain. One of the ideas we have is, we generate lots of metagenomic data, or what you call amplicon sequencing data for these marine fungi from select tourist beaches. and we generate it for 5 years, 10 years, then connect it with our GPS satellite data. That can we predict the change in the microbial communities or fungal communities over the period of time?
Yeah.
Maybe because of change in salinity or maybe change in temperature. But for that, we need a baseline data. So that's what we have been thinking. If anyone else is interested in this idea, they can just go ahead and try to get some funding. That would be good for marine mycology in India.
So can I make some few points at the end of the talk on session? First of all, I thank Sharma sir, Sridhar sir, and Bhatt sir for the igniting lot of minds and having a very nice discussion. And thank you, Dr. Shenoy, for a very nice talk on marine fungi, telling us about the Indian history, the literature, and brief about the NIO, what NIO is doing, and also letting us know that other people can also collaborate and use the NIO resources through of sampling, through collaborations and writing to the NIO people. And I very aptly said that a separate culture collection for a marine fungi is needed because it's a different complete strategy for the preservation and studies is required as compared to the normal fungi, which are mostly geophilic and the normal fungi. Thank you also for sharing your studies on marine fungi and tar balls. Your points to focus, 10 points to focus, are a good look into for the policymakers, the project managers of the funding agencies, and I hope your talk reaches out to them. And we need to continue to work, otherwise the previous significant works of our stalwarts will go in vain if there is a gap between in the marine studies. And I agree with you of the interdisciplinary research idea that needs to be taken ahead. Thank you, Dr. Shenoy. Thank you for this talk.
Thank you, Dr. Rohit Sharma. First of all, getting involved with MycoAsia as a founder of CBEC. Number two, as you were involved in that our marine mycology online seminar, then you came up with the idea of publishing some of the interesting learnings in the form of a paper. So that was a trigger. That was a great encouragement that you came up with this idea, and I got involved. And along with Professor Sridhar, we took it to a next level with the help of some experts in the field like Professor Bhatt and Patricia, Dr. Patricia, Dr. Suri, Dr. Sunil Deshmukh. Many people contributed to that paper, even though they are not the authors of that paper, but many of the Professor Sharma, of course, gave me the review in 2 days. I pestered him and just gave me the review in 2 days, and he was very encouraging. And therefore, I consider him as my guru of marine mycology, and we'd like to have more people in marine mycology. How it will happen, I don't know, but I'm just being optimistic here. And thank you very much to all the experts present today, Professor Sridhar, Professor Bhatt, Professor Sharma, Devdatta, and some other people in the audience for your patience. I mean, not only the attending, giving you valuable insight, sharing ideas, and some good criticism also. That will help me define some of the ideas. Thank you. Over to you, Dr. Shenoy. Yeah, thank you.
I thank everyone, all the audience members for joining for the today's talk. I think we can conclude here and see you at the next session of lecture 11 series in the next one. Thank you.
Thank you so much. Bye-bye.